welcome to the presentation. This is a shared talk, as Heiner mentioned, between Andrea Schnepf from Forschungszentrum Jülich and myself as the co-speakers of this Open Call 2 project, crop responses under phosphate deficiency, integration of plant physiology and sensing and modeling. The students listed here on top, Lisa, Magda, Samiga, and Felix, they more or less just started. So we'll present you a lot of fresh ideas and also some actually preliminary results already that hopefully will get you into the idea of what we are going to do with this. And of course, we have important collaborators here that are listed: Amy, Marshall Cologne, who will hopefully visit us soon. Here in Bonn, Jürgen Gahl, Frank Kochelinger, Lasse Klingbeil, Sabine Seidel, Eduardo Sell, and there are some more that I will mention during the talk. Okay, so first of all, why do we actually care about phosphate deficiency? And so there's a number of reasons that I want to give to you here that you appreciate why this is actually quite important. So first of all, phosphorus is one of 14 essential mineral nutrients and is a key determinant of yield. Second, soil P is generally not very plant available. And so in a nutshell, basically, if you fertilize phosphate, a large fraction of that phosphate will be immobilized, will stay in the soil, but will not be taken up by the soil. And this has to, by the plant, sorry. And so a minor fraction only is taken up by the plant, the rest will remain or less, not very usable for the plant. And this has to do with a number of different fractions of phosphorus or phosphate in the soil, some of which cannot be taken up, some of which are just insoluble and can therefore not be taken up. Some of them might be soluble but cannot be taken up because they have to be mineralized. And I don't want to go into too much detail here, but it's a complicated chemistry in the soil that prevents phosphorus from being basically very plant available. And the plant, the phosphate species that is taken up is called orthophosphate. So that's the molecule that is shown here. And I actually put this in presentation mode, in the pointer mode here. This molecule, the different orthophosphate species that in this talk I will sometimes also just call or abbreviate PI for inorganic phosphate. Yeah. Okay. So now a third reason why we should care about phosphate is that global P deposits are limited. Yeah, and so 80% is shown in this graph. And so the prediction of how in the time scales, there's some controversy about how long this might be, these deposits, how long they might last for good mineable phosphate. So between 100, maybe 300 years, there's some controversy, but the deposits are basically concentrated. 80% of the global mineable resources are considered to be in West Sahara and Morocco, in Jordan, in China, and in Russia. And so this also creates maybe an unwanted dependency, yeah, and some risk for food security then at the end. Now, also a very important reason that maybe is the most important of all that we should care about phosphate is that phosphate is a strong pollutant of open water bodies. And the reason for this is that phosphate is actually a limiting, growth limiting nutrient in aquatic ecosystems, yeah. And this is probably also one of the reasons that the authors of the study, A Safe Operating Space for Humanity, that you might be familiar with, Röckström et al. In this 2009 Nature paper, they consider phosphorus together with nitrogen and the loss of genetic diversity as the prime basically risks that crosses already planetary boundaries and endangers our basically safe operating space for humanity. And this now was, of course, a study from 2009. This has been updated, so, and phosphorus again is even considered in this original study as more threatening, and actually all the misflow and the deranged biochemical flows of phosphorus are considered to represent a greater risk to planetary health as compared to, for example, climate change, which is really surprising, but also understandable because of this risk of environmental pollution. And still in the updated version, basically 2022 from the Stockholm Brazilian Center, this is actually still considered this way, that phosphorus is indeed a very represents, or the deranged biochemical flows of phosphorus represent a very high risk to planetary health. Okay, so this in basically in a short wrap up. So this then means also, of course, that in order to meet the UN Sustainable Development Goals, there's a lot of things to do, among others. Plants or breeders should increase crop P use efficiency, but also we should be able to more precisely diagnose P deficiency and then predict consequences for P deficiency to basically allow management decisions that will basically reduce the input of fertilizer and the depletion of natural resources. And so in order to address these, or in order to meet these goals, there's a number of questions that have to be addressed. So first of all, how do crops sense P deficiency? Second, what impact does P deficiency actually have on crop physiology? and how do soil micro-plant interaction contribute to P availability, and what are the consequences of P deficiency on plant structure. And I will go through these points because we will touch on them with a different sub-project of our basically project here, Open Call 2 projects. And then the last point, what are the consequences of P deficiency on plant function? Now, if we talk about physiology as the combination of and the sum of basically biochemical and biophysical processes underlying a certain phenotype, this physiology or these combinations of biochemical reactions is enlarged as a consequence of the flow of genetic information in a given organism, in a plant, from DNA to RNA to protein to metabolites, and then these metabolites that generate structures and so on, that then finally results in the phenotype. Now, just to put this in perspective, so if you think about DNA, for example, we have a mice plant, we know that we have approximately 40,000 protein coding genes. This results in, if we allow also splicing variants, in approximately 100 to 200,000 different RNA molecules, this distinct RNA molecules. This then also, if we allow protein modifications, yeah, like glycosylation, phosphorylation, and so on, in approximately 200 to 1 million distinct proteins, and then in a number of metabolites that is hard to judge, but we think it's probably more than 10,000, but it could be even just the tip of the iceberg because the analytics are still not there to measure all of the metabolites. And so importantly now, these basically, these different levels of the flow of genetic information, they are all basically regulated by the environment. This could be, for example, plus minus phosphorus treatment, this could be management, this could be many things, but basically the environment will impinge on this. And just to give you this example, and so this means that the environment can change DNA, chromatin, for example, structure, chromatin modification, it can change the transcription, the stability of RNA, it can stimulate translation or provide or influence the turnover of proteins, make post-translation modifications and so on, that will result in a lot of changes that are dependent on environment. And this then also results in the fact that you can have two identical genotypes that have, of course, very different phenotypes in different environments. Now, just to stay with this example of phosphorus, if we look at this, we see, for example, and this is now a combination also considering different plant species here, not only maize, that we estimate that there could be more than 2,000 transcriptional changes alone with the difference between plus and minus phosphorus alone. And this will result in more than 1,500 metabolite changes. Yeah. And so I want to bring this slide here, and there are some papers that you can look at. And I want to bring this slide because what I want to say is that it's actually very difficult if you just look at metabolomics or transcriptomics. It's very difficult to separate cause and consequence. So what is physiologically relevant and what is not relevant? And in order to make this basically, to make this assessment whether something is physiologically relevant and should be looked at, basically, then also maybe as a readout for the diagnosis of nutrient deficiency in this particular case, it's important to be able to pinpoint genotype to phenotype interactions. And to do this, you need to be able to ideally have two genotypes that are identical, but in one, you take out one specific metabolite that you think might be physiologically relevant, and you can test this. In order to do this, you need genetic variability. You need to be able to manipulate or to find the genetic diversity in populations and natural populations that will allow you this direct correlation between genotype basically and phenotype. So that's important. So it's overwhelming otherwise, and it will be hard to find physiologically relevant phenotypes. So now what I want to say, or what I show you in the next slide basically is basically a cumulative findings in model plants about phosphate sensing, where exactly this genotype to phenotype correlations have been very vigorously addressed and analyzed. And I will not go into the details of how these forward and reverse genetic basically techniques actually work. So now, why do we actually care about phosphate sensing? And so the idea behind this is a little bit that, you know, if we want to understand or if we want to diagnose nutrient deficiency, ideally non-invasively, remotely, it can be very challenging, and I will come to this back in my presentation. But the problem is that, or the idea is here that maybe we can learn from plants how they actually sense the deficiency or the nutrient, yeah. And if we can learn from that, maybe we can learn also then how to actually diagnose this remotely. This is the idea behind this. And so, in a simple way, one could think that maybe there is a root cell, and this shows an electron microscopic picture of a root here, where if we take a cross-section, we take a single cell, and maybe in the single cell in the plasma membrane, there is a receptor that then can detect phosphate and can decide, okay, is there enough phosphate or not? And even though this seems like an attractive model, nothing like this has been found so far. And it's actually a little bit more complicated. And interestingly, plants actually don't sense directly phosphate. They do this indirectly. And this is shown here. And this is cumulative work of many different laboratories. Also, we have contributed quite a lot to this. Some papers are listed here. And this is now work from Esther Wiemer, mostly that I'm talking about, the PhD student in the lab who's about to finish. And she contributed to this 2 papers on top here. And this shows actually that in a P deficient conditions, there's a transcription regulator very conserved in plants that will bind to specific promoter elements that then will allow the transcription of many different genes. They will encode for proteins, for example, that are secreted to mineralize phosphate from organic sources, that will be transporters and so on, that will sequester and take up phosphate from the soil and so on. So many different genes, remobilization of phospholipids and so on, that will help the plant to overcome phosphate deficiency. Now, under phosphate replete condition, there's something interesting happening, and that is there's a signaling molecule being produced, which is called, it's a complicated name, it's INSP8, we often call it in the lab slang IP8, and this molecule looks a little bit like this, yeah, it's an inositol ring with many phosphates, and so there's different stereochemistry to be considered, but it doesn't matter. So basically the signaling molecule is produced, and this then binds to a repressor protein, which is called SPX1, and this repressor protein then makes a conformational change. And once this conformational change is made, it will bind to the transcription factor PHR1 and will stop all of the expression of these genes. Yeah? And this is kind of interesting because it means that the plant actually doesn't sense phosphate, actually it senses this signaling molecule as a proxy for phosphate. Yeah? And so now the idea is, of course, also could we then take advantage of this to read out phosphate deficiency in a better way. Okay. Now this is all Arabidopsis, I should say, model plant that is very easy, and genetic tools are available to do all of this research quite easily. And this will be then looked at in this Open Call 2 project by Lisa Sander and by Magda Putzkova in Peter Dörmann's lab. And I will show a couple of slides how this will be actually approached. Now, first of all, I want to say that Esther went a little bit deep into this mechanism, how actually the signal molecules are produced. And I want to point out one particular thing, that this enzyme can use IP6, phytic acid, that some people that are interested in nutrient availability and human nutrition might be interested in, can convert this into IP7 in a very interesting way and reversible way. And this all depends on the level of ATP, the currency of energy in a cell. And what Esther found is that the phosphate status very strongly correlates with the energy status of a cell. And this enzyme, in contrast to all other kinases that we are aware of, and there are thousands of kinases in plants, different kinases, it can make a reversible reaction where it can actually convert this signaling molecule precursor for this proxy of phosphate back to IP6 and thereby generate ATP, so recuperate the energy basically. So basically this enzyme is not only providing the messenger and the signal for phosphate signaling, but also it serves as a sensor for energy status of the cell. Yeah? And I'll come back to this later when I talk about work that I think Amy Marshall-Colon will be able to do for us. So now how about crops? And so how sensing works? Yeah, and so we want to do this in maize and barley in this Open Call 2 project. And so I want to talk about sensing, but also then about physiological traits relevant for P acquisition and P use efficiency. And so first of all, this will be then worked by Lisa in collaboration with Van Kovelinger's lab. And this is basically taking advantage of a European sequence index reverse genetic mice mutant collection. This is a very long name. Basically, this is a collection of maize lines where we have individual transposon-induced mutations in individual genes. And this is not transgenic, yeah? So this is a natural, basically, mutator transposon that has been used to generate this population. And so now we can directly ask, because it's all sequence, it's a wonderful resource, basically, we can ask now, okay, if this sensor component is missing, you know, so and maybe just to give this short information, we have approximately 1,200 this mutator-tagged F2 families that are sequenced, and they cover approximately 16,400 of all annotated maize genes. That's not all, but it's a large portion, and that is currently still being basically further amplified, this collection. And so the idea is now that we can take these different components of the sensor system and basically the sensing mechanism that we know in Arabidopsis, and now look at them, how relevant are they for maize? Yeah, we take the autologous genes and then we see if they are basically relevant for phosphate sensing. And hopefully by this method, basically we are then able to actually understand better phosphate perception, phosphate sensing in maize also. Now the other approach basically is taking advantage, and this is done by Lisa and by Samika from Pangyu's lab, is taking advantage of a magic population. And so this is a complete different principle. Here we have actually a population of 132 sequenced or indexed basically maize lines that all come from a founder population of 8 individual founding maize land races that are very diverse, that come from different places in Mexico. And interestingly, and I come back to this, they're also on places where we have very different phosphate availability. And so basically here we can now ask if there's allelic variation and whether some of the players that we identified in the model plant, basically in Arabidopsis, also basically are relevant for phosphate use efficiency in maize. But also here we have the chance, of course, because it's basically an unbiased approach where we basically make a genome-wide association mapping and then can make QTL mapping, QTL linkage basically to certain traits. It's an unbiased approach that will allow us also to identify novel players in phosphate use efficiency in maize. Okay, and this is also just as a side note used for in silico evaluation of root trait, the same magic population of root trait adaptations of maize land races to the native pedoclimatic conditions by Guillaume Lobé with his student Adrian Heymans. And Andrea will say later a short word to this also. Now we have an alternative approach to complement this maize approach, basically to identify the sensory basically mechanisms in maize. And this is basically done by Peter Dörmer's lab with Magda Kutzkowska here. And this is using barley and a more targeted strategy where we have CRISPR-Cas9. You might have heard about this, where we have basically a very precise tool to individually deactivate certain gene functions by a basically small cut that then will be repaired in plants, but normally not functionally, and then will result in a loss of function. And we can also do this with all of the different players in order to understand what are the players then in barley. And these plants then are under current European regulation transgenic. So this has to be then in a laboratory climate chamber basically approach in order to understand what the basically phosphate sensing basically is here. So then also, and this is also done by Magda and already started, she started with this experiments already, hydroponic cultures, plus minus phosphorus, for certain days, and then basically investigate the responses to P deficiencies with respect to metabolome. And here there will be anthocyanins that will be measured, chlorophyll, secondary metabolites such as flavonoids, phospholipids, inositol pyruphosphates, these signaling molecules, storage compounds, oil protein, and a number of more molecules that I'm not going to mention here. And this will be then validated, of course, by experiments that we have in Dickopshof, where we also have this long-term fertilizer experiment with phosphate-deficient plants. And samples already have been stored for this and harvested basically some time ago. And now they can also go in this pipeline to validate if this basically, if these changes we see in hydroponic are also basically under field condition, they also replicate basically. Now, what I want to mention is that we also are interested, and this is of course a short wrap up, in the contribution of microbes to P availability. And this is a project from Samiga with Pang Yu together. And before I tell about her project, I want to shortly mention a work that he has also presented the seminar series, but just to make this in a small slide, that in this Nature Plant paper that was published last year, Pang Yu and Frank Kohler's lab, he could show that that under certain genotypes, so this is genotype specific, can under nitrogen deficiency secrete flavones that will attract a certain group of bacteria, these Oxalobacteriaceae, and these bacteria then in turn, they will exudate certain substances that will induce the propagation of lateral root growth. And this helps the plant to overcome nitrogen deficiency. It's a very elaborate, beautiful, basically co-evolution between certain genotypes and taking advantage of these microbes in a way that makes, of course, that increases nutrient use efficiency, could be used in the breeding program now to get into elite lines, basically to take advantage of the microbial communities that are out there to help overcome nutrient deficiency. And one very interesting and promising aspect of this is, and this is just a short summary again, is that if you look at transcriptome data from different root sections, and remember, the transcriptome is one part of the genetic information that will then flow into these phenotypes, you will see that basically there is a strong correlation between different root zones and root transcriptome, meaning different root zones have different functions, also with respect to secretion that then can be seen in the metabolome. But also this has actually consequences for the better diversity of these bacteria. So for the bacterial community structure in these different root zones, they're distinct and most likely because of these distinct basically activities of the root zone. And this is actually promising to think about that this basically correlation between the root function and the bacterial basically is actually probably meaningful and might not only be playing a role in nitrogen deficiency, but also in phosphorus efficiency. And there's nothing known. And this is actually what she's doing with the same population. And there she will basically make these genome-wide association mapping QTL linkage in order to identify whether certain microbial communities basically and certain genotypes can interact and increase phosphate use efficiency in that combination. Okay. And so then basically now the question is, how does this all help with what we actually want to do in PhenoRob? Yeah. So of course the allelic variants, the QTLs, can be directly used for breeding, but also the physiological relevant traits might be used to diagnose nutrient status with better accuracy. Yeah. So one idea is maybe can we measure these molecules that are using the plants themselves to signal phosphate deficiency. And I'm saying this because phosphate deficiency has phenotypes that are well described in textbooks and so on, rigid growth, symptoms such as anthocyanin accumulation in older leaves, because phosphate is phloem movable, can be taken out from older leaves to younger and so on. But often these symptoms are not very specific. Yeah. And they can be confounded, for example, also sometimes even with nitrogen deficiency. You can also and high light and stress, and so you will also have this anthocyanin accumulation. This is something we have also observed together with in the project that we're doing together with Jin Hui and Jürgen Gahl in CP2. Yeah, that sometimes it's very difficult to tell them apart. So then the other 2 points that I want to mention before I finish and give over to Andrea is that we can also use this knowledge to generate smart plants, and I will show you in a minute what I mean with this, but also to use the data that we obtain for weighted correlation network analysis and multi-scale models. Now, what are these smart plants? And I want to make this very shortly. Basically, there's a complicated genetics behind it, but it means that we have a genetic trick to visualize a certain stress. Yeah, for example, P deficiency with a color reaction in a specific tissue. Yeah, and this is a transgenic approach, I should say. And I don't want to go in any of the details how this color pigment, it's a color pigment that is natural in certain plant groups, in Caryophyllaceae, such as in sugar beet and Echinacea and so on. And this will be transferred into a crop, for example, into barley. And this will then ideally, for example, show that there's P deficiency in this specific plant. Yeah. And so the important part here is that the promoter drives this pigmentation. So if you use a phosphate deficiency induced promoter, only phosphate deficient plants will have this, let's say, nodule, the node here that will be stained red in this particular case. Yeah? And in order to find such specific promoters, we started this quite some while ago. This is another student in my lab, DFG funded student, Deschambel, basically, and this is supported by Frank Kohlinger's lab for the analysis. We started transcriptome analysis in defined hydroponic cultures with all nutrient elements. And so now we are digging through this data. This is still basically work in progress to identify specific transcripts that are only induced by nitrogen deficiency, only induced by phosphorus deficiency, and so on, in order to make these specific promoters that then can be used for plants, for these smart plants. Yeah. And what do we do then with this? So the idea is then that with the smart plants, the plants will report P deficiency to enable semi-supervised learning of artificial intelligence algorithms under controlled conditions. But then these algorithms will be used to detect stress in the non-reported plant in the field. Yeah? And this will be done in collaboration with Jürgen Gahl, but we hope also for more input from other people, because it's not only RGB images, it's probably also hyperspectral and so on. And this is work in progress because these plants take a while to be produced. Okay. And then also, and this is my last slide for hand over, all of these inputs basically that we have, yeah. So these specific regulatory components that define basically phosphate use efficiency, but also phosphate sensing, they can be used for weighted correlation network analysis and multi-scale models. Yeah? And we hope therefore input from and help from Amy Marshall-Coulon, who will hopefully visit us this late in the summer. And basically, so this will allow gene to phenotype relationships. Yeah? And this will require basically physiological phenotypes, but also the genetic and transcriptome data that we are currently doing. And this can be used then by her lab to describe impact of P deficiency on photosynthesis, but also to provide a platform to test in silico perturbations. And with this, Andrea, I hope I didn't stress my time too much. I think it's your time to go.
Both Amy Marshall-Coulon and also our group are part of this so-called crop in silico community, where we try to describe different processes at different levels of organization and different spatial scales, ranging from the molecule to the ecosystem level, and also bridging between these scales using models. Where the group of Amy comes from the genome level and moving towards the larger scales, for example, photosynthesis and other processes at the cell level, and going to larger scales from there, where the focus of our group and of the PhD project in this Open Call 2 project is the organ level, with links to the smaller and larger scales. And there I think we have a nice way of interaction with the group of Amy, in particular with the potential to replace empirical response functions at the organ scale with more mechanistic process descriptions. So next, please. And this is an example which comes from the group of Guillaume Lobet and his PhD student Adrian Heymans, where they derive root hydraulic properties from small-scale knowledge, where they compute root hydraulic properties from cross sections using the Mecha model, and then develop this hydraulic atlas of maize roots. And here you can also see that different zones of the roots have different functions which manifest themselves in different anatomy, which then results into different root hydraulic properties. For example, root radial conductivity, which will affect the root water uptake. And similarly, another example from our group also does upscaling from the root to the whole plant hydraulic architecture. So there are, so yeah, next please. This is just an example. So this is a slide I wanted to include because at the recent retreat that we had a workshop about modeling, and there a discussion what is actually mechanistic and data-driven models. So I would say on the one hand, we have a pure mechanistic model, we can call it white box model, which is based on our mechanistic, on our theory and knowledge about the system, and on well-established physical relations where no calibration is necessary. Where on the right-hand side, we have data-driven models which are empirical and trained and have different input and output signals that are measurable, and then develop these can be also very complex models, but they don't give us an idea about the causality between the input and the output signals. Where in reality, we might have different levels or shades of gray in between these white and black box models, where there's also the idea that using a hybrid data-driven and mechanistic model approach could also overall have the benefit of the relative cost of information that is needed, both in terms of the physical information or the experimental information. And I discussed at the retreat with Christian Baukage that the way we can use this there are so many different options to use this that the limit is probably only our own imagination. Yeah, so next slide, please. Yeah, so I try to be very fast because the time is already short. So one way to use a combination of mechanistic and data-driven models is that to just produce synthetic data with a mechanistic model that can be used to train data-driven models, with the advantage that there will be more data outside of the experimental and observed range, but then that these data-driven models are then faster than a mechanistic model that can then be part of decision support tools that could be part of, for example, robotic applications. Working examples exist within PhenoRob, for example, in the group of Thomas Heckeley, and outside of PhenoRob, also for surrogate models, for example, for crop models. So next, please. Yeah, so how are we on time, Heiner? Just will you cut me short in a minute, or
1 minute? 1 minute left.
1 minute, okay. Then this will be the last slide. So this is the modeling approach of the OpenCall2 project, which I would classify as a light gray model, where we use functional structural plant model with empirical parameters. And these empirical parameters, for example, are observed from the plant structures to then parameterize our root architecture model. Next, please. Where we have, for example, images where we can segment the different structures out. Next, please. And please start the video also. Yeah, also in temporal information.
How do I get out of my pointer pen?
Okay, well, as we have so little time, then maybe we just skip it.
Sorry about this.
So the idea is to then extract relevant, next please, relevant information. And there we have for the roots automated pipelines to parameterize our root architecture models. They work surprisingly well also for the shoot part, but here we see high potential to collaborate, for example, with CP1. So this will be my last sentence. And from these structural informations, we will compute local response curve functions where individual specific parameters could be a level or will be a level of the function of the P level. And this will allow us to predict different structures at different P levels, and from there to do the functional modeling in terms of how this affects the, for example, root water uptake, nutrient uptake, photosynthesis, and so on. And yeah, I think we need to stop here because of the time, unfortunately. Okay, thanks.